So forensic is a science of solving crime to find the right person who has committed the crime, and as well as prevent those innocent people who are um, innocent victims, and they're still in prison. And there are many cases, there are a list of cases in which molecular biology or molecular biology techniques have helped to rescue the innocent and find the real criminal. So molecular biology plays an important role. And why it plays an important role is that our DNA, we all are made up of 46 chromosomes, or 23 pairs of chromosomes. There are protein coding genes which are very similar to each other, but the non-coding genes are very variable, and that variation in non-protein coding genes generates a probe-like feature to find unique signatures, and those signatures helps to identify the criminal. So it's a very systematic procedure that has to be followed. And if you have done wet lab this semester, molecular biology or molecular biotechnology, it's exactly the same thing. And there are many TV serials, there are many movies, they have shown the power of DNA in solving complicated crime, as well as in those cases where innocent has been you know uh, so finding the sample is very important, because at the crime scene, if we don't identify the right object that needs to be pursued for research, then we can't come to the right conclusion. So these samples could be hair follicle, uh, saliva, body fluid, skin cells, many cells, blood. So these objects serve as a potent material that gives direction to solving the mystery of crime and to identify the real uh, suspect for legal action. So reaching to the crime scene and identifying the site and identifying those objects that could be of interest for further investigation. So this person is wearing gloves, right? So let's say he or she is an agent. She's an agent. She's not using hand, mainly because not to destroy the evidence, because our hand could contain various nucleases, and those nucleases can degrade nucleic acid. And vice versa, if something is there poisonous on these things, then it will not affect the scientist or the agent or the laboratory worker. One thing conceptually I want you to understand is that there are 4 biomolecules, mainly protein, carbohydrate, lipids, and DNA and RNA. So let's take an example of an egg, right? Egg, you have breakfast, you prepare, sunny side up or omelette. Once a protein, egg, such as egg, is heated, it cannot come back, as an original egg. So protein denaturation under high temperature or high pH is irreversible. Proteins, if change shape, they don't come back easily. Lipids, fat, if you burn fat in heat, it cannot come back to the original chemical composition. Same goes with carbohydrate, let's say glucose. It's DNA is the only biomolecule. DNA is the only biomolecule that if heated, it can become single-stranded. And then when cooled, it can become double-stranded. So DNA is only molecule that can separate and then become double-stranded. Also, DNA is very stable. It can stand and very high degree of heat. So even if there is a fossil that is buried under the ground for thousands of years, DNA could serve as a point. Proteins in a dead person, proteins will be lysed very quickly. But DNA, so the value of DNA in forensics is immense, right? So even a hair follicle that has one cell intact could be enough to get to the bottom of the case. So you can ask me, how come one cell could be enough? So, remember PCR technique? You can amplify that DNA into million copies and then sequence. So one follicle, one cell of skin, one skin cell, saliva could be enough to get to the right person. So that's the scientific basis for narrowing down to the DNA. But I just want to change the topic a little bit. The most growing field of forensic these days is digital forensics. And so, for example, computer exams, data analysis, database, malware analysis, hacking, mobile devices, network analysis, messages, video analysis. Many things are going on in social media, which are permissible, which are not permissible, right? So so those are another growing field of forensics, the digital world, where in growing times, digital forensics will be very important, right? Uh, all the bank accounts are electronic now. Who knows what's going to happen tomorrow if there are people who are there to secure a website or a bank site, there are people who wants to break it. There's always this constant. Who knows, something can happen one day. But if there is a forensic in digital forensic in place, it makes you feel, we always want to go to a secured site rather than insecure site. But I believe that security is a growing process. As internet speed is becoming faster, there has to be more gadgets that would check hacking, bullying, malware attacks, so and so forth. So that's digital forensic is one of the growing area of forensic. So yeah. So digital forensics is a branch of forensic science encompassing the recovery, investigation, examination, and analysis of material found in digital devices, often in relation to the mobile device and a computer crime. So a phone that is with somebody for 24/7, if somebody is a victim, phone can take pictures. There can be important messages, right? Last minute messages. There could be a continued message for 1 year, some kind of mental harassment and so on and so forth. So digital DNA is important for molecular forensics, and digital is a growing field where it could relate to mental, social, society, psychology. All these can come around and can be, somebody is alone but continuously getting, wrong messages, stressful. Who knows what can happen, So so these, especially I'm taking example of phone because phone is just become our next buddy who knows everything about us, right? So it's an important source of important thing for forensic object for forensic investigation. So a typical forensic lab would look like any typical lab, just like on third floor of biology department, room 305 or 302, with these hoods, these kits, and various pipettes and these plastic wares and tubes, right? This is typical refrigerator and all those things. So there's nothing different a forensic lab would. Yes, but what is needed, that what is important here is there should not be any contamination, right? If there's a contamination, then the case will go in totally different direction. So the word forensic is derived from the Latin word forensis, meaning forum, a public place where in Roman times senators and others debated and held judicial proceedings. It is the application of science to those criminal and civil laws that are enforced by police agencies in criminal justice system. So this was the definition for forensic at the starting point. But now the definition is application of the tools of science as well as specific scientific facts to help solve the legal problems, right? So definition changes, it becomes very specific. What's the goal? To determine the determination of the cause, location, and the time of death, right? So to find the, of course, to get the justice, but at the same time, the person should be accountable who did something wrong. So forensic, as I showed you, that it involves lab workers. So there has to be a method in place, and this method can be automated to make it develop into a technology, and this technology can be applied to solve a case. And all those techniques that we have discussed until now can be used for forensics, PCR, DNA sequencing, and so on. So a little bit here and there, terminologies changes that I will discuss a little bit here. So identification of sample, preservation, preservation because to get the right data, to make it sure that there is no doubt, impeccable conclusion. Then analysis of data. I'm sorry. Documentation, and definitely one has to present the data. When you present a data, there are other people who look at the data and critique the data to make sure there are no loopholes, that you cannot again commit a same mistake that put an innocent person behind the bars. So the case has to be presented so that if there are any ambiguous questions, then it has to be addressed so that the finger points to the criminal 100%. So there are various stages: investigation preparation, evidence acquisition, analysis of evidence, or processing of samples using tools or technologies, then results, right? And then presentations. Another, as this molecular forensic was growing, what people understood that there are some genes or some DNA that doesn't code for a protein. Most of the protein coding genes are very much conserved in humans that cannot be used to probe a criminal crime scene, or to locate identify a criminal. So but what people realize that there are some unique signatures in the non-coding DNA, and these sequences were identified. So it's in the beginning, in 1900s, blood group typing was taken as a potential for identifying the criminal. Then protein isoforms testing were developed. Most important developed for DNA profiling was in 1980s by Alec Jeffries. And together with PCR, it brought a revolution in the field of forensic science. Then STR came into existence. Tandem repeats, short tandem repeats across the DNA. The short tandem repeats could be different in me and different in many of the human beings, and those short repeats can be used to identify. Then 13 CODIS core were identified. So it's a growing process to develop tools that can give convincing finger pointing to identify the criminal. Right. So DNA profiling is also called as DNA fingerprinting. It's the process of determining an individual's DNA characteristic. DNA analysis intended to identify a species rather than an individual is called as DNA barcoding. So let's say if there is a blood that is mixed with something, and there are many species, let's say a house where there are pets, and one has to identify where it's human or pet, so that their DNA barcoding becomes important. So this was discovered by Sir Alec Jeffreys in 1984. DNA profiling is a forensic technique in criminal investigation, comparing criminal suspects' profile to DNA evidence so as to assess the likelihood of their involvement in the crime scene. So as I mentioned to you, non-coding DNA, non-protein coding DNA becomes important. These are called as short tandem repeats. The system of DNA profiling is used today is based upon PCR and uses simple sequences. In UK, they have a DNA 17 loci system. So in US, it is CODIS. CODIS stands for Combined DNA Index System. Is the US National DNA Database created and maintained by FBI. CODIS consists of 3 levels of information: local DNA index system, then state DNA index system, and then national DNA index system. So all these data banks are being prepared. Then Y chromosome analysis to recognize, if a male is involved in crime scene. Mitochondrial DNA is important to see because we inherit mitochondria from our mother. So if there is any paternity suit, then Y chromosome typing and mitochondrial analysis could be very important in solving maternal disputes. So the basic process is to isolate the DNA, then quantitate the DNA, PCR, and then through electrophoresis, try to find out the criminal. Another very classical technique for solving paternity suit is RFLP, where the DNA is treated with restriction enzyme. So if an individual is homozygous, then the DNA has a profile like this on agarose gel. If a person is recessive, then you have this kind of profile, only one band. If somebody is heterozygous, you have this. So these serve as a standard. And then you take parents, you take kids, and disputed inheritance. Then one can, through these patterns, can sort out. It can go another step by doing southern blotting, by transferring into nitrocellulose paper that we have discussed in earlier chapters. So this could be the DNA profile of blood. This could be suspect 1, suspect 2, suspect 3. So you see that the blood that was gathered at the crime scene matches with the suspect 3. So this is a powerful technique too. And the most cells or the objects that are in demand to solve a case is skin cells, blood, hair, and saliva. So this was a small chapter that we finished.